When talking about natural selection, we're going to talk about 3 different modes of selection and what that can look like depending on each of the scenarios. So the first one that we have is directional selection, and you can think of this as the word directional. We are moving in one direction. So in directional selection, we are going to have the increase of expression of an extreme version of a trait in a population. So we're going to look at some examples, but first you can see over here, if we're looking at population and the outsides are one extreme, middle is the most common. In directional selection, we are going to move in one of these directions where over here is now going to become more common. So if we look at an example of this, most of the moths had light colored wings, but dark moths started to appear. Because moths lived against tree bark years later, most of the moths were dark. And so dark moths survived, creating more dark moth population, which means that over time we have moved in this direction towards one extreme. The next type we have is disruptive selection. And this is a process that is going to split a population into 2 groups, and it is removing the individuals with average traits in favor of the 2 extreme traits. So if we look at our bell curve over here, normally we would have this distribution down the middle with most common being just the average middle type. But in disruptive selection, we're now selecting for these 2 ends. So we're selecting for these 2 extremes, and they are going to become more common. So if we look at an example of this seen in nature, snakes that live on rocks are gray and will survive. Snakes that live on grass are green and will survive. But if we have an intermediate coloring, this could be disadvantaged because it is more visible to predators. So this is an example of why we would want to select those 2 extremes rather than the middle scenario. And the third type of selection we have is stabilizing selection. And this is going to eliminate the extreme expressions of a trait where the average expression leads to a higher fitness. So when the average is the best or the more beneficial trait, we're going to select for that, which is going to get rid of these narrow portions out here. Where there's less people, it's going to become even less common. And so if we look at an example, we can talk about human babies. So human babies born with below normal or above normal birth weights have a lower chance of survival than babies born with average birth weights. Therefore, birth weight varies little in human populations. So over time, because the survival is so low for the low end and the high end, it continues to select for this middle range, making it more and more common. We are now going to look at a few different examples and try to determine if it is directional, disruptive, or stabilizing selection. I know this isn't in your notes, but it's just going to be helpful for you to understand this now before we move on. So the first example we have is 2 different colorations of a species of beetles are distasteful to birds. Now we have to keep in mind, being distasteful to your predator is going to be a good thing for you. So we're going to want to select for those 2 opposite ends and not for the middle. And so this is going to be disruptive selection. It is going to be selecting for those 2 extremes. Our next example of brightly colored fish mate more frequently than neutral colored fish of the same species. So we're going to look for those extreme rare fish rather than the average ones. So we are going to be moving in one direction. This is directional selection. And a third example we have is the average baby chick weighs 45 grams when born. So the average is going to be towards the middle. We're going to want to normally be in this range, so it is stabilizing. And the last example, we have fossil evidence reveals that over time, the size of horses has been increasing. So if we're moving towards one direction, that is going to be directional. So when talking about whatever type of selection, we're going to be moving in some direction, or some alleles are going to become more common than others. So the allele frequency is that each allele has a frequency in a population's gene pool. And so our allele frequency we can calculate as the number of times an allele appears in a population, which essentially means it's how common that allele is. So essentially, The higher the allele frequency, that means that the greater the allele is there. So it's going to be more common the more frequent we see it. It also shows us how frequently the allele, usually dominant or recessive, appears within the gene pool. So it can show us how common it is and whether it's dominant or recessive. And remember that the gene pool is our combined alleles of all individuals in the population. So we can actually physically count the allele frequency. So we're going to calculate them, and P is going to represent our dominant allele and Q is going to represent the recessive allele. Don't ask me why P and Q were chosen. Those are just the letters that we use. So in any given population with only two versions of a gene, which is what our allele means, so you could have big B or little b, the dominant is going to be represented as P and the recessive version is going to be Q. So our equation looks something like P plus Q equals 1. So in our first example here, so in a population of wildflowers, we have a red allele as big R and the white allele as little r. And so if we have 500 total plants in our population, 20 of those plants are little r white, and 320 of them are homozygous red. And if you remember, homozygous, homo means same, so big R, and 160 are heterozygous, and hetero means different, so big R, little r. So we now want to calculate, we want to know how many total alleles for the flower color there are in this actual population, and how many dominant alleles and how many recessive alleles. So first off, we can just already tell that there is going to be 1,000 alleles total. There's 500 plants, each plant has 2, making a total of 1,000. But from there, we are going to want to divide this up into its subsections. So if we first focus on our little r, we have 500 total, 20 of them are little r. That's times 2, so it's going to be 20 plus 20 for each of those little r's. And we also have 160 of them that are big R, little r, which means we have 160 little r's. So if we add this together, that is going to be 40 plus 160, so we have a total of 200 little r's. If we do the same thing for our big R, we are going to have 320 that are big, so that's times 2 again. So we have 320 plus 320. And then again, we also have this 160 that have a big R present there for the heterozygous. So that is plus 160. Little harder math here. So we have 320 plus another 320. So that is going to give us 640 plus 160 is going to make 800, giving us our total of 1,000, as we said. So looking at this same example, we have our same wildflower population, 500 plants, same entire scenario. But now, instead of just figuring out how many alleles of each type we have, we want to find the allele frequency for the dominant and recessive alleles in the population. So if we start with our big R, we want to find the frequency. And we always want to use decimal place when finding frequency. So we had 800 out of 1,000 for our big R, which is going to be a frequency of 0.8. And if we remember, our dominant letter is our P. And for our little r, we had 200 out of a total of 1,000, which means that our frequency for this is going to be 0.2. And our lowercase letters, our recessive alleles, is the Q in our equation. And the reason I like math is that a lot of times we can check ourselves. So in our original equation, we have P plus Q equals 1. So here I can add 0.8 plus 0.2 and it equals 1. So we know, yes, this is in fact correct.